right now we use non-renewable resources. We use fossil fuels for transportation, and those supplies are getting limited, and we're trying to figure out ways that we can make new sources of this instead of going to fossil fuels. So we're interested in the process of making alkanes, which can be used for fuels or feedstocks in plants. And we saw a paper a couple of years ago where someone had identified an enzyme that makes alkanes. But a couple of people have studied this, and when they did, they found that the enzyme was like an old car. It turns over a couple of times and then stops. Okay, so this was a real problem. To study the enzyme in the test tube in the lab, we needed to add a series of other components to make rather a complicated reaction mixture. And when we added hydrogen peroxide in place of these other components, the reaction didn't turn over at all. And we were expecting, based on the way that peroxide can activate other enzymes, that it would actually substitute for those components. So when we realized that it suppressed the reaction instead of enhancing it, we wondered if hydrogen peroxide itself could be the problem that was causing the enzyme to turn over a couple of times and then stop. Okay? So we know of another enzyme called catalase that takes the hydrogen peroxide and converts it back to oxygen and water. So we added a little of this to the reaction, and lo and behold, we found that reaction didn't turn over 3 times, but just went on continuously for many, many hours. So instead of adding the catalase to the alkane-forming enzyme separately, we figured that we would make a fusion, a single protein containing both parts of this. We put it into E. coli just to test how this would work. It gave between 5 and 10-fold increase in the amount of alkane produced. The idea is now to transfer this into plants, whether it be algae or higher plants. They do a lot more electron transport, which means that we expect there to be a lot more hydrogen peroxide around. So we think that the effects that we see in E. coli are going to be dramatically better when we use photosynthetic organisms.